Good afternoon, everybody. I'm Xiaoying from Department of Neurology, Shanghai Hospital. It's my great honor to present my recent work about Hepcidin-Mediated disruption of brain iron metabolism in the pathogenesis of cognitive impairment in postmenopausal women. It is known that the incidence of cognitive impairment in postmenopausal women is significantly higher than that in men in the same age, and we can see it in this figure. The effect of estrogen supplement remain inconsistent, and not all postmenopausal women experience cognitive deficits. Considering the adverse effects, estrogen therapy for cognitive impairment is not the optimal approach. Menopause, marked by a decline in ovarian function and estrogen levels, results in the cessation of menstruation, which causes the active loss of iron in women. This reduction in iron loss leads to an accumulation of iron in the body, and emerging evidence suggests that excess iron may contribute to cognitive dysfunction. Therefore, identifying key molecules through which estrogen influences cognitive impairment may represent a critical avenue for preventing cognitive decline in postmenopausal women. Estrogen is closely related to human iron metabolism. After menopause, estrogen levels decreases, but brain iron deposition increases, and we can also see in this figure. Estrogen deficiency induces brain iron deposition by upregulating hepcidin expression in aged female mice. It was recently published in the literature. Hepcidin is a peptide secreted by the liver that regulates iron by binding to ferroportin. Its FPN1 is very important on the cell membrane, inducing its internalization and degradation, resulting in reduced cellular iron release and intracellular iron accumulation, which subsequently causes a series of damages. Within the damages, the most important one is ferroptosis. It is related to the iron accumulation. Ferroptosis is a form of cell death induced by the peroxidation of PUFA in cell membrane in the presence of ferrous iron, leading to lipid peroxidase and causing membrane damage. In the central nervous system, oligodendrocytes, OL, and their precursor cells, OPC, are highly susceptible to ferroptosis. Because of the reasons, the first one is OLs are primary iron storage cells in the CNS for myelin synthesis. And OPC exhibit developmental stage-specific overexpression of transferrin receptor 1, another important protein, TFR1, to enhance iron uptake. And because of lacking of antioxidant defenses such as GPX4, GSH, so OL and OPC are very poor to detoxification of lipid loss. And the last reason is high PUFA content in myelin membranes makes them prone to lipid peroxidation, leading to the demyelination and white matter lesions. Our previous clinical study have demonstrated that both chronic and acute white matter injuries, such as cerebral hyperperfusion and corpus callosum infarction, can lead to cognitive impairment with iron deposition in the white matter. Furthermore, in vascular cognitive impairment mice model, BCAS model, alleviating OPC injury and promoting remyelination have been shown to improve cognitive behavior. We used one is hyperforin, the other is minocycline. So our hypothesis is the decline in estrogen levels elevates hepatic hepcidin production, which subsequently induces intracellular iron accumulation. This iron overload triggers ferroptosis in OPC and OL, leading to white matter lesions and impaired remyelination. This pathological cascade is a critical contributor to cognitive impairment observed in postmenopausal women. We conduct a two-phase investigation. The first one is clinical cohort. Serum hepcidin levels, brain iron deposition, cognitive performance were correlated in postmenopausal women. The second one is preclinical study. OVX rats received subcutaneous hepcidin inhibitor GSHP or VICO and cognitive function, myelin integrity, ferroptis markers for analysis. In the first part, in the clinical study, the postmenopausal women were categorized into CI, cognitive impairment, and NC, non-cognitive impairment groups, based on whether their Montreal Cognitive Assessment MoCA scores were over or less than 26. In this study, we can see from the two tables, the table 1 is comparison of clinical baseline data in postmenopausal women matched groups with or without cognitive impairment. The baseline is almost the same. And the second table is comparison of the laboratory results of postmenopausal women in the matched two groups. And Hepcidin, MDA, and LPO increased in the cognitive impairment group. It showed serum hepcidin and oxidative stress levels correlated with cognitive impairment in postmenopausal women. And the serum hepcidin level, MDA level, and LPO level all have a negative correlation with MoCA score. And after the multivariated logistic regression analysis, we found that high serum hepcidin level is identified as an independent risk factor for cognitive impairment in postmenopausal women. This is our MRI results. We use a QSM processing workflow as below. And we divided the patients into two groups. One is low serum hepcidin group, the other is high serum hepcidin groups. Compare the two groups, we found that the high hepcidin group exhibit significantly elevated mean magic susceptibility, MMS. It indicates the iron deposition in the brain. In the following white matter tracks, left thalamic radiations and corpus callosum radiations and the other. No fiber showed reduced MMS in the high hepcidin group. And we also use a hippocampal segmentation map to evaluate the hippocampal relative volume. And we found that hippocampal relative volume decreased in high hepcidin group with elevated MMS and a decreased MoCA score. And there is also a positive correlation between hepcidin and white matter lesion volume. The second part is a preclinical study. We use the OVX rats as animal model. 9-week-old female rats underwent OVX or sham surgery followed by behavioral testing 8 weeks post-surgery. Compared to the sham group, OVX rats exhibit decreased estrogen levels, uterine atrophy, and impaired cognitive performance in the Y-maze and the Morris water maze test. And from the ELISA and WB results, the elevated hepcidin levels in both serum and brain tissue of OVX rats were accompanied by increased TFR1 and decreased FPN1 levels in brain tissues, suggesting iron metabolism dysregulation. At the same time, elevated AAC-SAR level and decreased GPX4 expression in OVX rat brain indicate ferroptosis. Prussian blue stain revealed iron deposition in both the corpus callosum and hippocampal regions of OVX rats. The blue granules indicate staining positive regions, and we can see that the number of Prussian blue positive granules indicate iron deposition was significantly increased in the corpus callosum and hippocampal region CA1, CA3, and the dentate gyrus of OVX rats compared to the sham group. And the fast blue LFB staining revealed myelin damage in the corpus callosum of OVX rats, and the IOD value of LFB staining in the corpus callosum was significantly decreased compared with the sham group. And the transmission electron microscopy TEM revealed tightly wrapped myelin sheath in the corpus callosum of sham rats, whereas OVX rats exhibit severe demyelination and loosely packed myelin structures. MBP immunofluorescence and Western blot analysis indicate that myelin damage in the hippocampal regions in CA1, CA3, and the dentate gyrus. And double immunofluorescence staining of oligo-2 red and GPX4 green in the hippocampal region of rats. We can see the quantitative analysis of oligo-2 and GPX4 collabed cells in the CA1 regions as a figure. OVX rats exhibit significantly reduced GPX4 expression in hippocampal oligodendrocytes, suggest oligodendrocytes for rhabdosis. And we use the TEM reviewed mitochondrial morphology in the hippocampal region of both groups. OVX rats exhibit mitochondrial shrinkage and increased membrane density. GSHP, it is a strong inhibitor of hepcidin. We administered the hepcidin inhibitor GSHP via subcutaneous injection to OVX rats starting at week 4 post-surgery and continued daily until week 8 post-surgery to assess the effect of hepcidin suppression on cognitive function in OVX rats. Y-maze and water-maze results showed GSHP alleviates cognitive behavioral impairment in OVX rats. And from the Western blot analysis results, we can see the hepcidin and some iron metabolism-related protein in hippocampal tissues of 3 rat groups. There is a decreased hepcidin, increased FPN1, and decreased TFR1 in the OVX group, and in the OVX plus GSHP group, the hepcidin level decreased, and the FPN1 level increased, and TFR1 level decreased too. And there is iron deposition in the OVX group, and we can see that in the OVX plus GSHP group, the iron deposition decreased in the hippocampus. Through the hippocampal MBP immunofluorescence stain, corpus callosum LFB stain, and WB analysis of MBP levels, we demonstrated that GSHP alleviates demyelination in OVX rats. TEM further revealed denser myelin structures in the corpus callosum and hippocampal region of OVX plus GSHP rats compared to OVX controls, suggesting that hepcidin inhibition ameliorates white matter injury in OVX rats. As demonstrated by Western blot and ELISA analysis, GSHP regulates phoroptosis-related protein in the serum and brain tissues, including upregulating GPX4 and GSH expression, while downregulating MDA, ACSL, and ROS levels. And from the double immunofluorescence staining of oligo-2 and GPX4, revealed a significant reduction in oligo-2 plus GPX4 collabo cells in the hippocampus of OVX rats as before, whereas the GSHP-treated group exhibit increased collabo cells, suggesting that GSHP alleviated phoroptosis in hippocampal oligodendrocytes. TM reviewed mitochondrial membrane damage, reduced mitochondrial volume and structure disorganization in the hippocampal and the corpus callosum region of OVX rats, suggesting phoroptosis. In contrast, the GSHP-treated group exhibit preserved mitochondrial integrity with clearer structure preservation. Here are the conclusions. Serum hepcidin levels correlated with cognitive impairment severity and serve as an independent risk factor in postmenopausal women. Elevated serum hepcidin level in postmenopausal women is associated with brain iron deposition and the severity of white matter lesions on MRI. Estrogen deficiency in OVX rats induces elevated serum and cerebral hepcidin levels, upregulates TFR1 and downregulated FPN1, leading to cerebral iron accumulation, oligodendrocytes phoroptosis, demyelination, and cognitive impairment. Inhibition of hepcidin expression with GHSP significantly reduced iron deposition in white matter, suppressed oxidative stress in the brain, and promoted remyelination in white matter regions, and significantly improved cognitive function in OVX rats. This study reveals that the hepcidin-mediated brain iron metabolism dysfunction critically contributes to the white matter lesions and cognitive impairment in postmenopausal women, while highlighting its potential as a therapeutic target for prevention. And I have a special thanks to our process team. Process is for French. The meaning in English is precise. What is process is prevention of cognitive impairment study. So we have a cohort study team, a clinical neurophysiology team, and a preclinical study team. This work was done by Dr. Wang Nuo and Dr. Gu Zhengsheng. Thanks for listening. That's all. Thank you.